Welcome back again. We are now going to look at how ARVs work because we want now to introduce a new class of ARV drugs in South Africa. So it's quite important that as we introduce the class, we look at all the classes of drugs and how they work and where this new class of drugs fits in. So just to orientate you a bit, what you have here in, blue purplish color, it's a virion, or what we would call an HIV virus. And in green, we have a cell, a white cell that has certain receptors which are important for HIV infection. We call them your CD4 receptors and your CCR5 core receptors. These cells are called CD4 cells, most of which are T helper cells, right? So just to narrate the story for you, what you have is an HI virus, right, that has certain proteins on its surface, GP120, GP41. Inside you have the viral nuclear capsid, right, which has 2 strands of RNA and certain enzymes, right? Mainly of importance is 3 enzymes: reverse transcriptase, we have integrase, and the third important enzyme is what we call protease, right? So the first step of HIV infection is what we would call attachment. Attachment happens when HIV glycoproteins attach to receptors that are found on the membrane of our white cells, right? This is where GP120 and GP41, these are glycoproteins, the HIV glycoproteins, would attach to the CD4 receptor and its core receptor CCR5, right? As soon as attachment has happened, which is the first step, this would be followed by fusion, where the 2 membranes, the cell membrane of the virus and the membrane of our cell, fuse, allowing direct communication between the 2 cytoplasm, right? And this is where now the viral genome or genetic material would be released into the cytoplasm of the cell. And when we say the viral genome or genetic material, we mean those 2 single-strand RNAs together with those 3 key enzymes, right? In the cytoplasm, an enzyme called reverse transcriptase would then convert viral RNA, this single-strand viral RNA, into a double strand or a DNA, viral DNA, right? There are drugs that are meant to inhibit this enzyme. They are called reverse transcriptase inhibitors. We've got those that are nucleoside and those that are non-nucleoside, right? So we've got nucleoside reverse transcriptase inhibitors. This would include drugs like zidovudine, tenofovir, lamivudine, emtricitabine, and abacavir. These are known drugs. These are drugs that we have been using in South Africa for years, and they are part of our guidelines. We also have non-nucleoside reverse transcriptase inhibitors. The main 2 ones that we need to know about is efavirenz, previously known as Stockrine, and also nevirapine, which is one of the drugs which we are likely, to stop using going forward, or use it very minimally, if at all. There's other second generation NNRTIs, rilpivirine, delaviridine, and so on. But the most important ones are efavirenz and nevirapine. Going back to our story, we have an RNA, a viral RNA, which is transcribed to a viral DNA by reverse transcriptase, and there are drugs that inhibit that. They are called reverse transcriptase inhibitors. This once we have this double strand DNA, it would be transported into our nucleus, where there's going to be a process of integration, where the viral DNA is integrated into the host or the human DNA, if you want to put it like that, right? The process of integrating viral DNA into the host DNA is facilitated by an enzyme called integrase, right? There are drugs that would block or inhibit integration from occurring. These are called integrase inhibitors. And dolutegravir, which is the main drug we want to talk about, would be part of those drugs. So we are introducing a new class. It's called an integrase inhibitor class. And one of the drugs which we have an interest in is dolutegravir. There's others: raltegravir, cabotegravir, bicitegravir, eltegravir, but the main one, which is going to be our focus as part of these new guidelines, is the introduction of dolutegravir, right? Once integration has happened, there's going to be translation of that captured DNA to be translated to RNA, to precursor proteins, and then to the formation of new virus, right? As part of the assembly, there's going to be, long strands of proteins, or what we call multi-protein chains, which require to be broken down or cleaved or cut and allow for the maturation of these budding virions, such that they can become matured and they can go and infect, more cells, right? That process of cutting, cleaving long strands or these multi-polyprotein chains is facilitated by an enzyme called protease, right? So we've got another drug class which are called protease inhibitors, and this would include drugs like lopinavir, ritonavir, atazanavir, duranavir, right? The commonly used protease inhibitor is a combination formulation, tablet or solution, that has lopinavir and ritonavir. So it's lopinavir and ritonavir. The brand names would include Calitra or Alluvia. If a patient is not taking any of these drugs, so there will be attachment, we'll have fusion, we'll have reverse transcription of the single-strand RNA to a double-strand DNA. The DNA will be transported to the nucleus, where integration would happen, and there'll be formation of new baby viruses that mature. And then this cycle just continues, destroying our white cells and creating more viruses that are going to infect more cells. And this is how you get high viral loads and low CD4 counts. And the process just continues like that. So integrase inhibitors are a new class being introduced in the country or in South Africa as part of the public health program. Their main function is to block the integration of the viral DNA into our host DNA. If this integration is blocked, replication cannot proceed, and therefore the virus would not be able to survive, and it will remain undetectable. There's quite a number of these drugs, but our key focus drug is dolutegravir as part of the integrase inhibitors. So without ART intervention, you would get integrase facilitating the integration of the viral DNA into the host DNA. And generally, if this process is allowed to continue, it then establishes a lifelong infection, right? If you introduce integrase inhibitors, when the viral genome DNA is supposed to be integrated, a drug like dolutegravir would then block the insertion of this DNA into our host DNA, and the process of replication cannot proceed beyond that point, and we would be very happy, and our patients would remain suppressed. I hope this was very helpful. Also try to watch the video showing how this actually happens. It's a nice video. Thank you very much. I hope just give us feedback as you go through these materials, and let us know if how is it going. If you have any questions, we are happy to answer. Thank you very much.